It's really great to be here in person, so I just wanted to thank ARM for helping to organize this. It's really inspiring to see you all together here. This conference has long been one of Aldevron's favorite conferences to collaborate at. So thank you again for organizing. My name is Tom Foti, and I'm the president of Aldevron's Protein Business Unit. I'm based in Madison, Wisconsin, and our headquarters with Aldevron is in Fargo, North Dakota. Looking forward today to share a little bit about what we're up to recently and a case study around cell and gene therapy that I wanted to share with you. Several of my colleagues are here this week, so look for them and look forward to refreshing some old friendships and some new partners. It's been an exciting year for Aldevron in 2021. On August 30th, Danaher Corporation acquired Aldevron, and we're happy to be part of Danaher's life science platform. On this slide, you'll recognize some brands that are well known in the industry. And today, and together, we want to offer our clients products and services both from research to preclinical all the way through commercial stage manufacturing for cell and gene therapies and for vaccines. We just started our transition activities with Danaher, and we're getting to know them, and we're also getting to know the operating companies within Danaher, and we're looking forward to combining efforts where it makes sense to better serve the market. So who here has some anxiety about supply chains? I was in conversations this morning and watching different talks, and manufacturing capacity and supply chain constraints is a major topic that everyone's trying to address. And we've seen historic supply chain challenges from the car industry to the home appliance industry and also in the biotech industry, and it's affected us in many ways. And the image on this slide is a congested seaport, and nowadays this has been common. And part of the problem, the challenge is that plasmid DNA, metaphorically, could be on one ship, Cas9 could be on another ship, LNPs or mRNA could be on yet a third ship. And getting that all organized is very complicated. The number one challenge we run into with our clients is not necessarily figuring out the complex biology that they're trying to address or come up with new innovative ideas. It's often around supply chains and trying to deliver to patients that need this material quickly and accelerated timelines is something that we want to partner with our clients to really figure out if we can continue to improve that for them. Manufacturing the central dogma of molecular biology has been foundational to Aldevron's strategy and strategic vision. And we believe we can do this also and simplify the supply chain. As you can see here in the graphic, in the red hexagon, we offer DNA, supercoiled plasmid DNA, and linearized DNA that is often used in cell and gene therapies, and many of you are well aware of Aldevron's plasmid work. But as many of you know, with messenger RNA and the success in the field recently using a polymerase, you can take linearized plasmid DNA and transcribe that into mRNA. And we've been able to collaborate with a lot of our mRNA clients because we manufacture proteins, and the third hexagon in green there highlights our protein business. The central dogma has been known for a long time. However, the complexities in manufacturing these products at large scale is very difficult, and also across multiple quality grades. Our DNA services have been foundational to our business. And since its inception, when Michael Chambers and John Ballantyne started Aldevron in 1998, we've, for the last several years, have been building the business, and since 2009, our protein business, and more recently, we've accelerated our mRNA manufacturing services out of Fargo, North Dakota. Our clients want high-quality material. However, we believe we can simplify the supply chain by offering these three manufacturing platforms, but it's just not the manufacturing capacity. It's the QC testing, the analytical packages that wrap around our delivery of these products that are also critically important. In addition to that, our clients require support from a quality assurance standpoint so that when they file their INDs and they're working with the FDA, they are able to gather the correct information, and we're happy to provide those services in a strategic way to our clients. Now I wanted to move in and talk a little bit about a case study, and the goal of many people in this audience, and we heard with our recent speakers here, is to develop cell therapy programs that are high quality, cost effective, and that can reach large patient populations. Again, a critical element to achieve this ambitious vision is a strategic supply chain. In the image on the left depicts a group of healthy donors, and in the middle are pooled cells, and you've seen many examples and conversations about this, and you're familiar with this, where people are knocking out specific genes, and here specifically we're knocking out the native TCR or major histocompatibility gene and knocking in a specific TCR for a tumor type. If these gene-modified cells can be expanded and scaled to potentially reach thousands of people, an allogenic therapy could be possible. 5 years ago, we had a vision of being able to develop and manufacture products that could support the cell and gene therapy so that they could produce large batches of gene-modified cells to help accelerate their rapidly expanding therapy programs. And in 2017, we were the first company to launch research-grade through GMP CRISPR enzymes. And since then, we have also offered a first company to offer GMP RMPs. The CRISPR enzymes we've offered as a standard product that is off the shelf. We heard also about off-the-shelf products as it relates to trying to simplify and standardize the supply chain. And this is something definitely that our clients appreciate. Along the way, we have learned a lot about supply chains. We have learned a lot about stability studies, product formulation, QC testing, and we continue to listen to our clients and think about how we can incorporate all these learnings into a better product. One client that we have collaborated recently with is a company called Vascugen. I would like to thank and recognize the team at Vascugen for sharing this data. They are a cell therapy company in Madison, Wisconsin, that has standardized their gene editing protocols using Aldevron's SPI5 Cas9 nuclease. Carter Cliff is the CEO at Vascugen, and you can reach out to him specifically if you have questions about their process. But in this slide, you can see a flow plot verifying that 91.6% of the cells have been properly gene knocked out. Vascugen introduced this in-process analytical tool, which you often see in the manufacturing process prior to their cell selection and cell expansion. The data suggests that the SPI5 Cas9 is active at knocking out the targeted gene of interest. Moving to the next slide, to confirm Vascugen's method for gene knockout was robust, they used SPI5 Cas9 nuclease in several of their manufacturing batches. Each of the bars represent a independent manufacturing batch, and so NF13 through N21. The data represents edits on the same cell line, knocking out the same gene, performed on different days with different operators. So it's just reassuring to see that the robustness of the process is working for Vascugen using Aldevron's SPI5 Cas9 nuclease. As you can see in their process, it's robust with 87.9% on average of their target gene over 9 batches that were tested. We're well below their target knockout spec indicated by the dotted line. When talking to Carter Cliff and the team at Vascugen, they mentioned that manufacturing gene-modified cells is a complex process, and the ability to have a turnkey solution off the shelf with Aldevron's SPI-Fi Cas9 nuclease is very helpful for them to advance their program. So I started this presentation talking about supply chain challenges that everyone is experiencing today. At Aldevron, we can offer a potential solution. We can manufacture and supply DNA, mRNA, and recombinant proteins from the same vendor at scale across various quality grades. I mentioned earlier that Aldevron was the first company to offer GMP RNP services so that clients could take a single reagent that is released in a GMP environment and then deliver that directly to their cell editing event. Last month, we were happy to announce a milestone in the company where we're the first company to release GMP RNPs and deliver that to a client. So in closing, at this conference, we have several of my colleagues are here. We would welcome an opportunity to talk about any of these programs with you. And with that, I'd be happy to take any questions, and thank you very much for your attention today. And lunch is coming, so if there's no questions, we can stay on track here. Okay, thank you.
Yeah. Is there a question?
Oh, hello.
Yeah, apologies.
Oh, here.
I'm curious. So congratulations on the GMP RMP.
Speaking of
Oh, yeah, congratulations on the GMP RMP release. I think a lot of us know that's crucial to getting CRISPR-based therapies into the clinic. I'm curious what you think about other nucleases. Cas9 is quickly becoming your granddaddy's nuclease due to IP and other issues. Can you speak to other nucleases that you're planning on offering in a GMP way?
Yeah, so we are able to offer what we call standard, and you're right, there's been a lot of enzyme evolution that has occurred over the last several years. SPI-Fi Cas9 is a slightly evolved enzyme that is different than wild type, that has low off target, so clients do appreciate that. Of the 10, let's call it 10 to 20 RNP kind of conversations that we're having, all those clients are looking at SPI-Fi. But we're a custom CDMO, so we can manufacture whatever the client wants us to make and then use our platform manufacturing and RNPs to produce and release different types of nucleases. Of course, we would have to go through the work of validating assays with a specific enzyme and a specific guide and target site, but some of the other validated assays that we have already in place for the RNP platform could be readily used. So we would have a head start there. So we can use off-the-shelf enzymes, or we're also bringing in new enzymes all the time that we're taking a look at. So hopefully that answers your question, and I can talk to you after if you want.